Have you ever stared at a cut on your finger, or maybe seen a bruised bone eventually mend, and just thought, how did my body do that? It's almost like, well, like magic, this self-repairing trick.
It really is something, isn't it?
Yeah. So today, we're taking a deep dive into something truly fundamental, yet profoundly intricate: wound healing.
Mm-hmm.
Our mission for this deep dive is to pull back the curtain on the incredible, intricate choreography of how our bodies mend themselves. We're talking from the tiniest cells you can't see right up to the bigger changes you clearly witness.
Yeah.
And this isn't just for, medical experts. It's for anyone who's ever wondered about the astonishing processes just humming along inside us, keeping us whole.
And it really is a wonder. Think about it. Our ability to repair tissue after an injury or surgery is, well, it's an ancient foundational ability. Our bodies evolved over eons.
Right. Survival depends on it.
Injured organisms simply wouldn't survive without it. So understanding the science behind this, the chemical signals, the cellular armies, the whole molecular dance, it gives us a crucial roadmap. It helps us figure out how to best help the body heal. And honestly, it's the key to developing new cutting-edge biological approaches to make that healing even better.
Okay, let's unpack a foundational concept then, one that might surprise you. When we talk about healing, we often just lump everything together, don't we?
We do.
But there's apparently a huge difference between true regeneration and simple repair. What's the core distinction there, and I guess, why does it matter so much?
Okay, yeah, the core distinction really comes down to perfection, or the lack of it. Regeneration is when your body restores tissue that's structurally and functionally indistinguishable from the original.
Like it was never even hurt.
Exactly. It's like building a perfect replica. Bone is a fantastic example. It truly regenerates.
Yeah.
Your liver also has this remarkable capacity.
Wow.
But repair, on the other hand, is when tissue integrity is reestablished primarily through fibrotic scar tissue.
Ah, scar tissue.
Think of scar tissue as your body's emergency spot weld. It's strong, sure, but it's coarser, less organized, and has fewer cells than the native tissue it replaced.
So it's functional, but not perfect.
Right. And here's the kicker. With very few exceptions like bone and liver, most tissue disruption in our bodies results in this kind of repair, not true regeneration.
So it's about the quality of the fix then, a perfect rebuild versus a sturdy but maybe visible patch job. That makes a lot of sense. And speaking of patches and rebuilds, not all cells in our body are equally good at dividing and regenerating, are they? We have different kinds of cellular players on this team.
Precisely. We categorize them by their proliferative capacity, their ability to divide. You've got labile cells, like those in your outer skin layer or the lining of your mouth.
The ones that are always turning over.
Exactly. They're constantly dividing throughout your life, always ready for action. Then there are stable cells, like fibroblasts, the main builders of connective tissue.
Okay.
They usually have a very low duplication rate, kind of quiet most of the time, but they can rapidly wake up and proliferate in response to injury.
Like when you break a bone.
That's a great example. When you break a bone, specialized cells called mesenchymal cells quickly differentiate into osteoblasts and osteoclasts, the bone-building and bone-resorbing cells, to get that healing going.
Right.
Conversely, you have permanent cells, like your specialized nerve cells and cardiac muscle cells. They simply do not divide after birth. Once they're gone, they're gone.
And I imagine that really impacts what we expect from healing clinically. You can't just, grow a new heart muscle easily.
Exactly right.
Yeah.
A surgeon's expectation for normal healing has to be realistic based on the tissue. A fibrous scar is perfectly normal and expected for a skin wound, no problem there. But that same type of fibrous scar would be considered suboptimal, maybe even a failure, in bone healing because bone can regenerate.
Wow.
It just highlights how exquisitely tailored our body's responses are to different tissues.
That brings us nicely to the macro level view of healing, which is often described by its intentions. It's almost like the body has different strategies depending on the situation. We talk about healing by first intention, second intention, and even a clever third intention.
Yeah, these terms basically describe the conditions under which a wound closes. Healing by first intention is the ideal scenario. Think. A clean cut, like a surgical incision, that's closed immediately, maybe with stitches.
Neat and tidy.
Exactly. The edges are neatly brought together, it heals quickly with minimal scarring, and the edges don't pull apart. But if conditions are less favorable, say an injury that tears away a lot of tissue, an avulsive injury, or if there's infection, or it just can't be closed neatly,
Then it's second intention.
then healing occurs through second intention. Here, the body has to slowly fill the tissue defect with new granulation tissue and connective tissue from the bottom up. It's a much more drawn-out process.
Makes sense. It's a bigger job.
Right. And then for very complex or contaminated wounds, surgeons sometimes use a third intention approach. This is basically a staged procedure.
How does that work?
Well, the wound is cleaned up thoroughly, debrided, and then left open to heal by second intention for about 5 to 7 days. This allows it to form healthy granulation tissue and ensures any infection is controlled.
Okay.
Once it looks healthier, it's then surgically closed, effectively shifting it to heal by first intention. It's a smart way to give the body the best possible shot in tricky situations.
That's fascinating, like a strategic multi-phase plan. Very clever.
Yeah.
So whether it's by first, second, or third intention, the body generally follows a universal script, right? Especially for skin wounds.
Pretty much, yeah.
Let's introduce the 3 distinct overlapping phases of healing. I like to think of it as a beautifully choreographed drama, the inflammatory, proliferative, and remodeling phases.
Indeed, it's quite a performance, act or. The inflammatory phase. This is the body's immediate response, its first responders rushing to the scene.
How long does this usually last?
Typically about 3 to 5 days. It kicks off with rapid constriction of the injured blood vessels, a quick tightening, like a biological tourniquet, basically to stop the bleeding.
Hemostasis.
Right, hemostasis. Then a key molecule, factor 12, acts like a crucial spark, igniting this whole cascade of events that leads to clotting. Circulating platelets, tiny cell fragments, rapidly swarm the injury site.
Like little emergency responders.
They really are. They form a primary plug within a mesh of fibrin. Now, this clot isn't just about stopping the bleed. Critically, it provides a temporary scaffold, a provisional matrix, for cells to migrate through as the repair begins.
Right, so this initial clot isn't just a plug. It's literally sending out signals, like a tiny biological alarm system.
Exactly. You've got it. The clot also serves as a critical reservoir for powerful signaling molecules called cytokines and growth factors.
Okay.
These are released by the activated platelets, and they essentially broadcast messages to the surrounding cells, telling them what to do next. Come here, we need help, that kind of thing.
And what about the blood vessels after that initial tightening?
Good question. After that initial quick vasoconstriction, a more persistent widening, vasodilation, kicks in. This is driven by chemicals like histamine and prostaglandins. It increases the permeability of the vessels, allowing blood plasma and other cellular mediators to flood into the injured area.
And that's what we see, right? The swelling and redness.
Clinically, this is exactly where you see the classic signs of inflammation: swelling, redness, heat, and pain. It's the body's way of shouting, "Attention! Injury here!"
And then the cellular army starts arriving.
Yes, those cytokines act like homing beacons, recruiting the first wave of immune cells, the neutrophils.
The cleanup crew.
They're the first on the scene, arriving within minutes, flooding the site like a specialized cleanup crew. They release powerful enzymes, proteases, that break down damaged tissue, and they gobble up bacteria, clearing the debris.
But they don't stick around long.
They're short-lived, yeah. Unless there's a serious infection, their numbers typically peak and then decline after a few days. But the pro-inflammatory cytokines they release, like TNF-alpha and IL-1, continue to fuel that inflammatory response.
Okay, so neutrophils fade. Then who comes next?
As the neutrophils diminish, another crucial cell takes center stage, the macrophage.
Ah, the macrophages. I've heard they're important.
Hugely important. What's truly fascinating here is their dual role. They continue the microdebridement, the fine-tuned cleanup, breaking down injured tissue and consuming cellular debris using enzymes like collagenases and elastases and through phagocytosis.
So more cleanup.
Yes, but even more importantly, they become the primary source of healing mediators. Once activated, they release a whole battery of growth factors and cytokines, TGFs, PDGF, IGFs, TNF-alpha, IL-1. The list goes on.
So they're directing traffic now.
They're basically the directors. Exactly. These signals amplify and perpetuate the healing cascade, influencing all the subsequent phases by regulating tissue remodeling, inducing new collagen formation, and kickstarting new blood vessel growth, angiogenesis. Macrophages aren't just the cleanup crew. They're coordinating the entire repair process.
Wow.
Seriously. Wounds that lack sufficient macrophages show defective repair. They are absolutely vital.
That makes perfect sense. They're not just cleaning, they're commanding.
Okay, that takes us to Act 2, the proliferative phase. This phase is stimulated by all those growth factors from Act 1, right?
Exactly.
Typically lasting from about 3 days up to maybe 3 weeks. And visually, this is where you start to see that characteristic pink granular tissue. It's like the body's new construction site is really kicking off.
Yes, that pink granular tissue is literally a sign of new growth, just packed with fresh activity. A crucial first step here is angiogenesis.
New blood vessels. That's right, the generation of new capillary blood vessels. Think of it as laying down new plumbing. This process is driven by the low oxygen levels in the wound hypoxia and by growth factors like VEGF and FGF2 that signal for new blood vessel growth.
Why is that so important?
Well, they establish a local microcirculation. You need that to supply the oxygen and nutrients needed for the really high metabolic demands of these regenerating tissues. They're working hard.
Gotcha. And who's doing the building?
Around the same time, matrix-generating fibroblasts, the main builders of connective tissue, migrate into the wound, again, responding to signals from those inflammatory cells, especially macrophages.
And they start laying down stuff.
They start synthesizing new extracellular matrix, the scaffold of tissues, and immature type 3 collagen. This forms a structure that supports those newly formed blood vessels.
Okay.
And it's a brilliant feedback loop, because these stimulated fibroblasts also secrete their own growth factors, which helps sustain the repair process.
And this makes the wound stronger.
Yes. The rapid deposition of collagen quickly increases the tensile strength of the wound. It means it becomes stronger and less reliant on, say, sutures holding it together.
And what about the surface of the wound? How does the body seal that off?
Good point. At the surface, reepithelialization occurs. New skin cells, epidermal cells, proliferate from the wound margins and effectively crawl over the wound bed to seal off the exposed area.
Does that happen at the same speed everywhere?
Interestingly, no. This process actually happens faster in oral mucosal wounds, like inside your mouth, compared to skin wounds.
Why is that?
Mainly because the mouth stays moist. This allows the epithelial cells to migrate directly over the clot instead of having to tunnel under a dry scab, which slows things down on the skin.
Ah, clever. And do they just keep growing?
Nope. Once the new skin edges meet in the middle, a phenomenon called contact inhibition halts further lateral proliferation. It's like they sense they've met and they stop moving sideways.
Wow.
And all of this is dramatically helped by wound contraction. This is driven by special cells called myofibroblasts.
Myofibroblasts? Like muscle?
Kind of. They're transformed fibroblasts that generate strong, muscle-like contractile forces. They literally pull the wound margins together, shrinking the defect. It's quite powerful.
Incredible. Okay, so we've stopped the bleeding, cleared the debris, started building new tissue, sealed the surface. But the process isn't over yet, is it?
Not by a long shot.
That leads us to Act 3, the remodeling phase. This is the long game, you said. It can extend for months or even years.
That's right. Potentially up to several years in some cases.
This is where that immature scar tissue truly strengthens and refines itself. And you said here's where it gets really interesting, because it's a constant tug of war.
This is indeed the long game of healing, yeah. A finely choreographed balance between breaking down old matrix and forming new. As the metabolic demands of the healing wound decrease, that rich network of new blood vessels that formed earlier, the angiogenesis, it starts to regress, shrink back.
Okay, less need for supply lines.
Exactly. And under the direction of various cytokines and growth factors, the collagen matrix is continually broken down, then resynthesized, reorganized, and stabilized by molecular cross-linking into a mature scar.
So it's constantly being worked on.
Constantly. Crucially, the fibroblasts that were so active begin to disappear through programmed cell death, apoptosis. And the weaker type 3 collagen that was deposited during that rapid building phase is gradually replaced by stronger, more resilient type I collagen.
Making the scar tougher over time.
Precisely. Correspondingly, the tensile strength of the scar tissue gradually increases. It eventually reaches approximately 80% of the original strength of the uninjured tissue. Never quite 100% usually, but pretty good.
And what controls this breaking down and rebuilding? You mentioned microscopic crews.
Yeah, this delicate balance of scar collagen and the extracellular matrix is largely regulated by enzymes called matrix metalloproteases, or MMPs. Think of them as the demolition crew in taking apart old structures. And they're balanced by tissue inhibitors of MMPs, or TIMPs, which basically make sure the demolition crew doesn't go too far.
Right, checks and balances.
Absolutely critical.
Yeah.
Because any disruption in this MMP-TIMP balance can lead to either an exuberant, overgrown scar, too much building, or not enough demolition, or to the wound falling apart, dehiscence, too much demolition.
So it's a constant refinement and strengthening process.
Fascinating. Now, while those three acts are a general model, healing isn't identical everywhere, as we discussed. Let's shift focus to specialized healing. Not all wounds are created equal. Let's start with nerve injury. That sounds tricky.
Nerve injury, particularly in areas like the face and mouth, can range significantly in severity. The mildest form, neuropraxia, is like a temporary functional block. Think of it like a kink in a garden hose.
Okay.
Nerve conduction is interrupted, maybe due to compression, but the nerve itself, the structure, is intact. Recovery is usually spontaneous, often within 3 to 4 weeks.
That sounds manageable. What's next? Next up is axonotmesis. This is more serious. The internal wires, the axons, are severed, but the protective outer connective tissue sheaths of the nerve remain intact.
So the cable's still there, but the wires inside are cut.
Good analogy. Recovery here depends on the extent of the internal damage and whether those axons can regrow down the intact tubes. Then there's neuromesis. This is a complete transection of the entire nerve trunk.
The whole cable is cut.
Exactly. Spontaneous recovery from this type of injury is quite rare. Surgery is usually needed to realign the ends.
So how does a nerve even attempt to heal after being severed? It sounds incredibly complex.
It is incredibly complex. Shortly after nerve severance, a process called Wallerian degeneration begins in the part of the nerve disconnected from the cell body. The axons break up and are essentially eaten or phagocytosed by the surrounding Schwann cells and infiltrating macrophages.
Clearing the path.
Exactly. Once this debris is cleared, the Schwann cells on the surviving proximal end start to grow outwards, attempting to bridge the gap and connect with the distal segment. They proliferate to form these structures called Bunger's bands.
Boner's bands.
Yeah, to think of them as biological guide tubes, forming a pathway for regenerating axonal sprouts that emerge from the proximal nerve stump.
Guiding the regrowth.
Precisely. These growing Schwann cells also release specific neurotrophic factors, proteins that promote nerve regeneration, and adhesion molecules that help direct the axonal growth.
What happens if the ends aren't lined up?
Well, if the nerve stumps aren't surgically realigned properly, these growing Schwann cells and axonal sprouts can get disorganized, kind of lost, and form a tangled mass called a neuroma.
And that can be painful.
Often very painful, yes. It's basically a disorganized knot of nerve tissue trying to grow but going nowhere. And the regeneration rate itself, it's generally considered to be agonizingly slow, approximately 1 millimeter per day.
1 millimeter a day. That really puts long nerve injuries into perspective.
It certainly does. Recovery can take a very long time.
Okay, let's turn to bone healing. You mentioned earlier this is a special case of true regeneration. That sounds amazing.
If we connect this back to our earlier discussion, yes, bone is biologically privileged in this way. It heals by regeneration, not just repair.
A true biological superpower, you called it.
It really is a remarkable exception. While most of our tissues settle for that spot weld with scar tissue, bone has this incredible ability to completely regenerate itself, structurally and functionally, as if the injury never happened.
How does that happen spontaneously, say, if you just put a cast on a fracture?
Right. If left alone, a fractured bone can often restore itself through what we call indirect healing. After the injury, a blood clot, a hematoma, forms. This immediately triggers an intense inflammatory response, just like in skin.
Okay.
This organizing blood clot acts as a scaffold for reparative cells. Pluripotential mesenchymal cells, those versatile stem-like cells, rapidly produce a soft fracture callus.
The biological splint you mentioned.
Exactly. It's made of fibrous tissue, cartilage, and immature bone, or woven bone. This splint binds the severed bone segments together and helps dampen motion at the fracture site, allowing bony union to occur. Eventually, this callus is remodeled into mature lamellar bone.
And what if a surgeon intervenes with plates and screws? That sounds different.
Yes, more commonly these days, especially with significant fractures, a surgeon facilitates direct healing, sometimes called primary bone healing. This is an abbreviated, essentially callus-free process.
How does that work without a callus?
It involves surgically realigning the displaced bone segments very precisely and then rigidly stabilizing them with internal fixation devices like plates and screws. This rigid stability is key. In areas where the fracture ends are touching, in direct contact, the fracture line is filled concentrically by mature lamellar bone, essentially knitting together directly. If there are small gaps, they get filled by a succession of fibrous tissue, fibrocartilage, and then woven bone, which then remodels. But crucially, if there's minimal or no micro-movement at the fracture site due to that rigid fixation, direct healing happens without any significant soft callus formation.
And then, just like skin, bone goes through its own remodeling phase. It sounds like a constant sculpting process.
Absolutely. Subsequent bone remodeling eventually restores the original shape and internal architecture of the bone. This functional sculpting is carried out by what we call the basic multicellular unit, or BMU.
The BMU crew.
The BMU crew, yeah. Think of it as a microscopic, highly organized bone construction and demolition team traveling along the bone surface or tunneling through it. You have osteoclasts, the demolition experts, excavating old or damaged bone.
Bacon tunnels.
Right. And right behind them, following in the tunnel, are the osteoblasts, the master builders, laying down fresh new bone matrix called osteoid, which then mineralizes.
What controls these BMUs?
They're controlled by growth factors, cytokines, hormones, and especially by mechanical signals. The physical stress or strain produced by functional loading, just using the bone, plays a significant role.
How so?
Well, low levels of strain, maybe around 2,000 microstrains, are actually necessary and beneficial for cell differentiation and callus remodeling. A little bit of stress is good.
But not too much.
Exactly. Too much strain, exceeding that roughly 2,000 microstrain threshold, can hinder bone formation. It can lead to fibrous tissue forming instead of bone, or even non-unions, where the bone fails to heal together at all. It's a delicate balance.
So the right amount of stress is good, but too much is detrimental. Okay, let's look at another specialized example, one many of us have probably experienced. Extraction wounds, like after a tooth is pulled. You said this is a classic example of healing by second intention.
Indeed. It's a great example. Immediately after a tooth is removed, blood fills the socket, and a clot forms, sealing off the torn blood vessels. Very important first step.
Right.
Within the first 24 to 48 hours, this clot begins to organize. Blood vessels around the remaining periodontal ligament remnants engorge, white blood cells migrate in, and a fibrin layer forms.
Then what happens in the first week or so?
In the first week, epithelial cells, the skin cells from the gum margins, start to grow over the surface of this organizing clot. Meanwhile, deeper down, osteoclasts, those bone demolition cells, begin to accumulate along the surrounding bone crest, preparing for some natural reshaping, and new blood vessel formation, angiogenesis, proceeds in the remnants of the periodontal ligament.
Okay, lots going on. What about week 2 and 3?
By the second week, the clot continues to organize with active fibroblast activity, fibroplasia, laying down connective tissue. New blood vessels penetrate deeper into the clot, and new bone, osteoid, slowly starts extending from the socket walls inward.
Filling in from the sides.
Exactly. By the third week, the socket is largely filled with granulation tissue and some poorly calcified woven bone forms at the perimeter. Critically, the surface is usually completely re-epithelialized by now, often with minimal or no scar.
So it looks healed on top fairly quickly.
Relatively, yes. But the active bone remodeling continues underneath for weeks and months. You don't even typically see radiographic evidence of significant new bone on x-rays until 6 to 8 weeks after the extraction.
Wow, that long.
Yeah, and the final healed bone structure isn't fully discernible radiographically for maybe 4 to 6 months. It's a slow process underneath.
What happens if that crucial initial clot doesn't form or if it breaks down? I heard of something called a dry socket. That sounds unpleasant.
That's exactly what a localized alveolar osteitis, or a dry socket, is. And yes, it is unpleasant. When that initial blood clot fails to form properly or it disintegrates prematurely, perhaps due to infection or trauma,
The whole process gets derailed.
significantly delayed, yes. Because that crucial granulation tissue matrix, the initial scaffold built on the clot, isn't present. It leaves the bone exposed in the socket, which is painful, and leads to a much slower rate of new tissue formation. Healing still happens, but it's much slower and more uncomfortable.
Okay, one more specialized example. Skin grafts. We often hear about full thickness versus split thickness grafts. What's the basic difference and how do they actually heal once they're put in place?
Good question. A full thickness graft includes the entire epidermis, the top layer, and the full dermis, the layer underneath. A split thickness graft consists of the epidermis and only a varying amount of the dermis, making it thinner.
Right. So how do they survive initially? They're cut off from their blood supply.
Initially, yeah. After grafting, the first nutritional support for a free skin graft comes from the fluid, the plasma, that oozes from the host bed's capillaries. It's called a plasmatic imbibition. A fibrin clot quickly forms between the graft and the bed, kind of like a biological glue, securing the graft in place.
But that plasma won't sustain it long term.
No, the graft's survival critically depends on a rapid reestablishment of blood supply from the host bed. This happens in 2 key ways, often overlapping. There's neovascularization, the actual ingrowth of new capillary buds from the host bed into the graft.
Like exploring roots.
Sort of, yeah. And inosculation, which is where existing blood vessels in the graft directly connect, or anastomose, with vessels in the host bed.
Linking up the old plumbing.
Exactly. Endothelial capillary buds from the host bed essentially tunneled into the graft, reaching the dermo-epidermal junction within about 48 hours.
And how do you know it's working?
Clinically, when you see the graft turn pink, typically between the 3rd and 5th day, that's your signal that these vital vascular connections have been successfully made. The graft is taking.
Okay. And then fibroblasts get involved?
Yes. Host fibroblasts, those building cells, invade that fibrin layer by day 4. By day 9 or so, new blood vessels and fibroblasts have created a pretty firm union, anchoring the grafts securely.
And how about sensation? Does feeling ever return to normal in a grafted area?
Reinnervation of the skin graft does occur. Nerve fibers from the surrounding host tissue enter the graft, usually from its base and sides, and they tend to follow the pathways of existing nerve sheaths within the graft dermis.
When does sensation start coming back?
Sensation recovery usually begins within about 2 months after transplantation. However, it rarely attains the full sensory quality of normal original skin. It's often diminished or altered.
Okay. And performance-wise, split versus full thickness, any major differences?
Yes, split-thickness grafts, being thinner, are generally more susceptible to trauma later on, and they tend to contract or shrink more as they heal. But they have a higher rate of initial survival or take because they're thinner and revascularize more easily.
Okay.
Full-thickness grafts, on the other hand, don't take quite as readily and are slower to revascularize, but they are more robust against trauma long-term and undergo minimal shrinkage, which is often better cosmetically. So pros and cons to each.
Fascinating trade-offs. Okay, so we've walked through this elegant dance of healing, from the first responders to the long-term remodeling and these specialized cases. But what happens when that choreography goes wrong? While healing is often uneventful, complications can definitely arise. Let's talk about wound infection first. That seems like a big one.
It is a significant issue, yeah. Wound infection typically results from a substantial bacterial presence in a susceptible wound. Now, it's important to distinguish infection from simple contamination. Pretty much all wounds pick up some bacteria.
Right.
But a true wound infection is generally defined by a bacterial burden of more than about 100,000 organisms per gram of tissue. At that level, the bacteria actively impair healing.
It's not just the number, though.
No, it's also about the pathogenicity, the aggressiveness of the bacteria, and, crucially, the host's immune response and tissue condition that determine if healing gets derailed.
How does infection actually stop healing?
Well, the constant presence of bacteria stimulates the body's immune defenses, leading to a persistent flood of inflammatory mediators. Neutrophils, our early immune cells, keep coming, and they release harmful cytotoxic enzymes and oxygen-free radicals.
Which damages healthy tissue, too.
Exactly. It can lead to thrombosis, blood clots in small vessels, hypoxia, lack of oxygen, and further tissue damage. This oxygen deprivation, combined with toxic bacterial byproducts, actually enhances bacterial growth and causes more tissue breakdown. Newly formed cells and delicate collagen are particularly vulnerable.
And clinically, what are the signs we look for?
You'll see the classic signs: erythema, redness, warmth, swelling, pain, maybe a foul odor, and often purulent discharge or pus.
What makes a wound more likely to get infected?
Several factors contribute. Inadequate tissue perfusion or oxygenation is a big one. Failure of aseptic technique during surgery, of course, excessive tissue trauma during the injury or surgery, the presence of necrotic, dead tissue, or foreign bodies.
Places for bacteria to hide.
Precisely. And, of course, a compromised host immune system. Prevention is really key. Meticulous surgical technique, careful handling, thorough debridement of dead tissue, good hemostasis to prevent blood collections, eliminating any empty spaces in the wound, and proper postoperative care.
Another really dramatic complication is wound dehiscence. That sounds bad.
It is serious, yes. Wound dehiscence is the partial or sometimes total separation of the wound margins. It typically shows up within the first week or so after surgery.
Is it usually because the stitches failed?
Actually, no. Most instances of dehiscence happen because the tissue itself fails to hold together adequately. The healing just isn't strong enough yet. It's usually a fundamental issue with the early healing process, not typically due to sutures breaking or being improperly placed.
Wow. Okay. And then there's the issue of scars that just don't settle down properly. You mentioned proliferative scarring, including hypertrophic scars and keloids. These sound like the body getting a little too enthusiastic with the repair process.
That's a great way to put it, yeah. Both hypertrophic scars and keloids share some underlying features. They tend to be hypervascular lots of blood vessels, hypercellular, too many cells, especially fibroblasts, and have excessive amounts of extracellular matrix components, particularly type I collagen. Plus, there's often persistent inflammation.
But they look different, right? How do you tell them apart?
They have distinct clinical differences, yes. Hypertrophic scars generally arise relatively soon after the injury, and critically, they stay within the original wound boundaries. They might be raised and red, but they don't spread outwards. And over time, they often tend to flatten and recede somewhat.
Okay. And keloids?
Keloids, conversely, often manifest months after the initial injury. The key difference is that they grow beyond the original wound boundaries, like claws extending outwards, and unfortunately, they rarely subside on their own.
Are some people more prone to them?
Yes, there's a strong genetic and racial predisposition for keloid formation. They're more common in individuals with darker skin types, and they frequently occur in specific locations like the face, earlobes, and the anterior chest or upper back.
What's believed to be the underlying mechanism for this overzealous scarring? It sounds like the body just doesn't get the signal to stop building.
Exactly. It's believed that altered regulation of programmed cell death, apoptosis, plays a significant role. Normally, apoptosis helps remove excess inflammatory cells and fibroblasts as healing matures.
Right.
But if this process is dysregulated, it can lead to this excessive scarring, persistent inflammation, and overproduction of all those building blocks. Both keloids and hypertrophic scars show a sustained elevation of certain growth factors, things like TGF, PDGF, IL-1, IGFI. Powerful GO signals, yes. They tell local fibroblasts to keep multiplying and pump out excessive amounts of collagen and extracellular matrix. Additionally, proliferative scar tissue often has increased numbers of factors that promote new blood vessel formation and also more histamine-secreting mast cells, which can further stimulate fibrous tissue growth. It's like the on switch is stuck.
Is there anything effective that can be done for them, especially keloids?
Unfortunately, there's no truly reliably effective therapy for keloids. It's an ongoing challenge. Common methods, like injecting corticosteroids directly into the scar, aim to inhibit protein synthesis, fibroblast proliferation, new blood vessel formation, and collagen production. But results can be variable, and recurrence is common.
Okay, so given all this complexity and the potential for things to go wrong, what can actually be done to optimize healing? What factors can surgeons and even patients proactively address to give the body its best shot? Let's start with minimizing tissue trauma during surgery. You mentioned that earlier.
Minimizing trauma is absolutely paramount. It directly promotes faster, better healing, and it really should be a central consideration at every single stage of a surgical procedure, right from planning the incision through to closure.
How so?
Well, a properly planned incision is just long enough for optimal exposure, no longer. Ideally made with one clean, consistent stroke. Sharp tissue dissection, rather than tearing, causes less damage. Careful placement and use of retractors to avoid crushing tissue. And using sutures judiciously.
Judiciously, meaning not too many or too tight.
Exactly. Sutures are obviously needed to hold tissues together until the wound is healed sufficiently. But too many sutures, or tying them too tightly, can actually increase infection risk by creating pockets for bacteria or strangling the tissue, cutting off its vital blood supply.
Less is often more.
Okay. And beyond minimizing trauma, we need proper hemostasis controlling bleeding and debridement, which you described as cleaning up the wound. Why are these so critical? Spot on. Complete hemostasis, stopping all bleeding, is crucial to prevent hematoma formation. That's a collection of blood or serum pooling in the wound site.
Why is a hematoma bad?
It creates an ideal breeding ground, a nutrient-rich medium for microorganisms. Plus, the pressure from a large hematoma can compromise blood flow to the overlying tissues, potentially causing them to die, leading to necrosis and dehiscence.
Got it. And debridement?
Equally important is debridement, the surgical removal of any dead, devitalized, or contaminated tissue in any foreign bodies. These things act like hideouts for bacteria, shielding them from the body's immune defenses and antibiotics.
Okay.
Necrotic tissue also actively reduces host immune defenses locally, encourages infection, prolongs the inflammatory phase, which we want to move past, and physically obstructs the process of contraction and epithelialization. Debridement literally reduces the microbial load, removes toxins, and gets rid of physical barriers to healing.
Makes sense. What about things like implants?
Yeah, it's worth noting that even prosthetic grafts and implants, although designed to be biocompatible, can sometimes act as foreign bodies and stimulate chronic inflammatory reactions that can adversely affect healing in the surrounding tissues.
Okay. Now, this raises an important question you touched on. How critical is oxygen when we talk about tissue perfusion, getting blood supply to the area? We often hear oxygen is vital, but why, at a fundamental level, is it such a big deal for healing?
Oxygen is profoundly critical. You could almost say the oxygen levels in your tissue, the oxygen tension, largely drive the entire healing response. Low tissue oxygen levels, hypoxia, severely inhibit healing.
How?
Well, cells switch to less efficient anaerobic metabolism, producing lactate, which lowers the pH, making the environment acidic, all contributing to tissue breakdown. Oxygen is an absolute necessity for countless healing processes. Energy production, making new collagen, the replication of fibroblasts in the endothelial cells that form new blood vessels, the ability of our leukocytes, our white blood cells, to kill bacteria effectively, and angiogenesis itself.
So pretty much everything.
Pretty much everything important, yes. When local circulation is impaired, whether from rough surgical handling, tissues drying out, sutures tied too tight, poorly designed surgical flaps, or underlying conditions like hypovolemia, severe anemia, or peripheral vascular disease, it hinders the delivery of vital nutrients, oxygen, and antibodies to the wound.
And even the immune cells need it.
Yes, even our immune cells, like neutrophils, require a certain minimal level of oxygen tension to exert their bacteria-killing effect effectively. Many of the healing problems we see associated with things like diabetes, radiation damage, and chronic infection can be traced back, at least in part, to this critical issue of local tissue oxygen deprivation.
And I imagine something like smoking would have a major, major impact here. That sounds like that double whammy we mentioned earlier.
Absolutely. Smoking is notoriously detrimental to wound healing, primarily because it severely decreases tissue oxygenation. The nicotine causes peripheral vasoconstriction, a tightening of the small blood vessels, and the effect from smoking just one cigarette can last for up to an hour.
An hour, wow.
Yeah, so imagine a pack-a-day smoker. Their tissues are effectively hypoxic, oxygen-starved for a significant portion of the day. Smoking also increases carboxyhemoglobin in the blood, which means the red blood cells carry less oxygen. It increases platelet aggregation, making blood stickier, increases blood viscosity, making it thicker, and directly decreases collagen deposition and the formation of other beneficial substances like prostacyclin.
So it hits healing from multiple angles.
Multiple angles, all bad for healing. For optimal patient outcomes, it's strongly suggested that patients abstain from smoking for at least 1 week, preferably longer, before and after any surgical procedures. It really does make a substantial difference.
Okay. What about chronic conditions like diabetes? How does uncontrolled blood sugar specifically impact wound healing?
Numerous studies have shown that the higher incidence of wound infection and impaired healing, often seen in diabetic patients, has less to do with the diagnosis of diabetes itself and more to do with hyperglycemia, that is, chronically high blood sugar levels.
So it's the high sugar itself that's the problem.
Primarily, yes. Hyperglycemia negatively impacts nearly every aspect of wound healing. It adversely affects the immune system. Things like neutrophil and lymphocyte function, their ability to get to the wound, chemotaxis, and their ability to engulf bacteria, phagocytosis, are all impaired.
Okay.
Uncontrolled blood glucose also hinders the flexibility of red blood cells and directly impairs blood flow through those critical small vessels, the microvasculature, at the wound surface. This leads to oxygen and nutrient deficiency right where it's needed most.
A vulnerable situation.
Exactly. This local oxygen starvation, combined with the impaired immune cell recruitment and function, makes diabetic wounds highly vulnerable to bacterial and fungal infections and just generally slow to heal. Good glycemic control is crucial.
And what about patients who are generally immunocompromised for reasons other than diabetes?
Yeah, anytime the immune response is significantly debilitated, surgical outcomes are potentially compromised. Healing relies heavily on a properly functioning immune system, especially in that initial inflammatory phase.
Is there a way to measure that?
One parameter sometimes used is the total lymphocyte count in the blood. Levels below about 800 cells per microliter are often considered indicative of a severe deficit, signaling a significantly weakened cellular immune response.
What kind of conditions lead to this?
Examples include patients with advanced HIV infection, those on long-term immunosuppressive therapy, perhaps after an organ transplant, or patients taking high-dose corticosteroids for extended periods for inflammatory conditions.
How do steroids specifically affect healing?
Steroids, like prednisone, can have pretty significant inhibitory effects. They depress fibroplasia, fibroblast activity, collagen formation, and neovascularity, new blood vessel growth. Fibroblasts are delayed in reaching the wound site. Overall wound strength can decrease by as much as 30%, and both reepithelialization and wound contraction can be impaired.
Is there anything that can counteract that?
Interestingly, some studies suggest that supplemental vitamin A can sometimes help attenuate or lessen some of these inhibitory effects of steroids on wound healing.
What about chemotherapy drugs?
Anti-neoplastic agents, or chemotherapy drugs, primarily work by interfering with DNA and RNA synthesis in rapidly dividing cells. This, unfortunately, also impairs the proliferation of fibroblasts and their ability to form collagen. Plus, many chemo regimens cause neutropenia-low white blood cell counts, which directly predisposes patients to wound infection.
Okay, let's talk about radiation injury. Especially in areas like the head and neck, where patients might receive therapy for cancer, that sounds like it could cause long-term problems for healing.
It absolutely does.
Yeah.
Therapeutic radiation, while targeting cancer cells, inevitably causes collateral damage to adjacent healthy tissues. This significantly reduces their long-term capacity for regeneration and repair.
Does the damage happen right away?
The pathological processes begin immediately, but the clinical effects might not become apparent for weeks, months, or even years later. Early acute changes involve the death of rapidly dividing cells, especially in high turnover tissues like the lining of the mouth, oral mucositis, or the skin, dermatitis. There's also a significant cytokine-mediated inflammatory response early on.
And the late effects, you said those are permanent.
Yes, the late effects of radiation are generally considered permanent and are directly related to the total radiation dose received. They typically involve changes like collagen becoming dense and stiff, hyalinization, widespread fibrosis, scarring, and chronic hypoxia.
Why the hypoxia?
It's due to something called obliterative vasculitis. Essentially, the small blood vessels in the irradiated area become progressively damaged, scarred, and narrowed, leading to a permanent reduction in blood flow and oxygen delivery. Tissue susceptibility to infection also permanently increases.
So healing in irradiated tissue must be really difficult.
It is very complicated. These late effects are irreversible. Wound dehiscence is common, and healing is often slow and incomplete. In severe cases, successful healing after surgery in a heavily irradiated field may only be achieved by surgically removing all the compromised non-vital tissue and covering the defects with a well-vascularized tissue flap, meaning tissue brought in from elsewhere with its own robust blood supply. It may even require removal of a vascular dead bone. It's a major reconstructive challenge.
Does age itself play a significant role? Do older people just heal slower?
Generally speaking, wound healing is indeed often faster in the young and tends to be somewhat protracted or slower in the elderly.
Why is that?
This apparent decline likely results from a combination of factors associated with aging. A gradual reduction in overall tissue metabolism, potentially decreased circulatory efficiency, an accumulation of damaged cellular components over time, and maybe the buildup of oxidative radicals.
But is it always a big difference?
Well, it's important to note that in the absence of other compromising systemic conditions, like the diabetes or vascular disease, we talked about the differences in healing solely attributable to chronological age can actually be quite small. A healthy elderly person may heal almost as well, albeit perhaps a bit slower than a younger person. Comorbidities often play a bigger role than age itself.
Finally, let's talk about nutrition. This seems absolutely fundamental. What we eat must have a massive impact on building new tissue.
Adequate nutrition is absolutely critical for normal repair. It's foundational. In malnourished patients, the formation of new connective tissue is delayed, angiogenesis is decreased, and overall wound healing and remodeling are prolonged. You simply can't build new tissue without the necessary raw materials.
What are the key building blocks? Protein.
Protein and amino acids are vital, yes, especially certain ones like methionine, histidine, and arginine. They are the literal building blocks for new cells and matrix proteins like collagen. Clinically, low serum albumin, a measure of protein in your blood below about 2 grams per deciliter, or low prealbumin below 17 milligrams per deciliter, are strong indicators of protein malnutrition and are associated with delayed healing.
Can taking extra protein help?
Glutamine supplementation, an amino acid, has shown some benefit in certain stress states. But it's important to remember that while a minimum protein intake is absolutely crucial, simply consuming a very high protein diet won't necessarily shorten healing time if you're already adequately nourished. You need enough, but excess doesn't necessarily speed things up beyond normal.
Okay. And what about specific vitamins and minerals? Are there some superstar nutrients for healing?
Yes, several vitamins and trace minerals play essential roles. Vitamin A is important. It stimulates fibroplasia, collagen cross-linking, which adds strength, and epithelialization. Can even help stimulate these processes in wounds that are slowed down by steroid use, as we mentioned.
Vitamin C. I hear that's important for collagen.
Vitamin C is absolutely essential for collagen synthesis. Hydroxylation of proline and lysine, key steps in making stable collagen, requires vitamin C. A deficiency not only impairs healing, but in severe cases like scurvy, can cause previously healed wounds to actually rupture.
Wow. Any others?
B-complex vitamins and cobalt are important for antibody formation and white blood cell function, supporting the immune response. As for trace minerals, magnesium, copper, crucial for collagen cross-linking via the enzyme lysyl oxidase, calcium needed for collagenase function and remodeling, iron, oxygen transport, and zinc are all important.
Zinc often gets mentioned for healing.
Zinc deficiency definitely slows down both fibroplasia and reepithelialization. However, it's also important not to overdo it with zinc supplements. Excess levels can actually be harmful by inhibiting macrophage migration and function, and potentially interfering with copper absorption and collagen cross-linking. Like many things in biology, it's about balance.
A very delicate balance indeed. Okay, so we've explored this incredibly intricate symphony of wound healing, the players, the acts, the potential complications, and the factors that influence it. But this increased understanding isn't just for diagnosing problems, it's leading to ways to actively modulate wound healing. So what does this all mean for the future? How are we moving beyond just watching healing happen to actually intervening and maybe even improving it?
Yeah, that's the exciting part. It means we're shifting from mostly passive treatments, like just dressing a wound, to actively trying to influence the wound microenvironment to promote better, faster, or more regenerative healing. Growth factors are absolutely central to this.
We talked about how important they are as signals.
Exactly. These are the powerful signaling molecules, the proteins, that orchestrate all those cellular activities like proliferation, migration, matrix synthesis, basically telling the cells what to do and when. So the clinical approach gaining ground involves applying these growth factors, often produced using recombinant DNA technology, directly to the wound.
Just put them on the wound.
Essentially, yes, often incorporated into gels or scaffolds. The big challenge, though, lies in figuring out precisely which specific growth factors to use, when in the healing process to apply them, and at exactly what dose or concentration for the maximum beneficial effect without causing problems. It's complex.
Can you give us some specific examples of these growth factors being used now, or maybe some promising new therapies based on them?
Certainly, one of the earliest and maybe most well-known examples is bacapamin. It's a recombinant human platelet-derived growth factor, rhPDGF.
PDGF, okay.
It was actually the first FDA-approved recombinant growth factor product specifically for treating chronic cutaneous wounds, like diabetic foot ulcers. It works by increasing fibroblast replication, boosting collagenase activity, which helps remodeling, and stimulating extracellular matrix synthesis.
Does it work well?
It can be effective, yes, but its very high cost has unfortunately been a significant barrier to its widespread use. Researchers are also exploring other factors, like recombinant human keratinocyte growth factor 2, KGF2, which has shown promise in accelerating healing in animal models.
What about for nerves or bones?
Yes, for peripheral nerve healing, naturally occurring proteins called neurotrophins, things like nerve growth factor, NGF, brain-derived neurotrophic factor, BDNF, and others are being actively investigated. They're normally secreted by Schwann cells and support nerve survival and growth, so applying them exogenously holds potential.
And for bone, where we saw that amazing regeneration potential.
Absolutely. In bone grafting procedures, certain osteoinductive growth factors are particularly exciting because they can actively promote new bone formation. This could potentially reduce or eliminate the need to harvest the patient's own bone from another site, which has its own risks and morbidity.
Which factors are key for bone?
The bone morphogenetic proteins, or BMPs, especially BMP2 and BMP7, have received the most attention. They have this remarkable ability to induce undifferentiated mesenchymal cells, those versatile precursor cells, to transform into bone-building osteoblasts.
Like flipping a switch to make bone.
Exactly like flipping a switch. One commercially available product, NSUSE bone graft, contains recombinant human BMP2, rhBMP2, delivered on an absorbable collagen sponge. It's been approved by the FDA for specific uses like spinal fusion, certain tibial fractures, and some oral and maxillofacial bone grafting procedures.
Sounds revolutionary. Any downsides?
Again, high costs are a significant factor. And there have been some safety concerns and controversies regarding potential side effects with off-label or high-dose use. So ongoing research is really focused on refining the delivery methods, clarifying guidelines, and perhaps developing more affordable or targeted formulations.
That seems to lead naturally into the idea of gene therapy. That always sounds like something straight out of science fiction, but how does it potentially play into wound healing?
Gene therapy for wound healing isn't sci-fi anymore. It's definitely an active area of research. The driving idea is to achieve very selective, controlled, and localized expression of a specific growth factor right where it's needed for a precise period.
So instead of just applying the protein, you tell the cells to make it themselves.
Exactly. Unlike a single dose of an externally applied growth factor protein, which might get washed away or degrade quickly, gene transfer could allow for targeted, consistent, and potentially high concentrations of these therapeutic healing peptides right in the wound environment, maybe for up to 2 weeks or longer from a single application.
How do you get the genes in there?
Various methods are being explored, including using modified, harmless viruses as delivery vehicles, viral vectors, or chemical, electrical, or mechanical methods to get the therapeutic DNA into the target cells at the injury site.
Has it worked in experiments?
Yes, experimental studies have already demonstrated success, for instance, in stimulating significant bone formation by delivering the human BMP2 gene using an adenovirus vector in rodent models. The potential is huge. This kind of technology could eventually lead to much less invasive reconstructive procedures where we can essentially program the body's own cells to become mini factories for the specific healing factors needed.
That's incredible potential. And finally, let's touch on dermal and mucosal substitutes, bioengineered skin or tissue replacements. How are these changing wound care and giving the body a helping hand?
Immediate wound coverage is often critical, especially for large wounds like burns. It protects the wound from dehydration, infection, and further mechanical injury, while autologous grafts using the patient's own skin remain the gold standard in many situations.
They require taking skin from somewhere else.
Right, which creates another wound site. So bioengineered substitutes are increasingly being developed and adopted as alternatives or adjuncts.
What kinds are there?
They can be broadly categorized. Some are based on cultured sheets of the patient's own epidermal cells. Others focus on providing a dermal component, like a collagen scaffold. And some are composite or bilayer products, aiming to replace both epidermal and dermal layers.
Can you give an example?
Sure. A well-known example is Apligraf. It's an allogeneic meating derived from a donor, not the patient living bilayer skin substitute. It's actually derived from cultured neonatal foreskin cells, keratinocytes and fibroblasts, grown on a collagen matrix.
Doesn't the body reject it if it's from a donor?
Fascinatingly, this particular product seems to be processed in a way that it doesn't typically elicit a strong immune rejection response. It's FDA approved for treating chronic wounds like venous leg ulcers and diabetic foot ulcers.
What's its main benefit? Just covering the wound.
It does provide coverage, but perhaps the chief effect of many of these advanced skin replacements isn't just passive covering. It's thought that they actively promote healing by releasing their own growth factors, and importantly, by stimulating the host wound bed to produce a variety of its own beneficial wound-healing cytokines. They act like a biological stimulus.
Kickstarting the host's own healing.
Exactly. The ultimate long-term goal for bioengineers in this field is to develop truly functional, engineered skin that contains all the necessary cell types and structural components to fully guide healing, allowing for wound closure with a substitute that perfectly replicates native tissue structure and function, and, crucially, dramatically limits or even prevents scar formation. Imagine a future where significant wounds could heal perfectly without a trace. That's the dream.
Wow. From that immediate life-saving platelet plug, through those intricate phases of inflammation and construction, right up to the years of subtle cellular and molecular remodeling, the sheer complexity and really the elegance of the body's wound healing processes are just astounding when you stop and think about it.
It really is.
And to consider the incredible strides science is making now, not just to understand it, but to actively assist this inherent powerful capability that exists within all of us. It's like our bodies are constantly, silently striving for wholeness, for perfection.
This constant drive for homeostasis, that internal balance and repair at the cellular level, is a truly remarkable undertow of our very existence. It's happening right now, in you, without you even having to think about it. So let me turn it back to you and our listeners. What stands out to you most about the body's innate, almost tenacious drive to repair itself, even when it's facing really significant challenges?
Something to definitely ponder. Thank you for joining us on this fascinating deep dive into the world of wound healing.